Hi everyone, welcome to your second lecture today. We're going to focus on biotechnology. I know that we have talked about chapter 13 or the concepts in chapter 13. This chapter was about the structure of DNA and the replication of DNA. That's what we mainly focused on. There is one more section left we have to talk about, and that deals in some of the biotechnology stuff that you may or may not see on the exam. We're going to do a virtual version of one of the labs that are involved in biotechnology this week. So this is again going to give you some background into those labs and those techniques. So obviously we know that the discovery of DNA was a huge deal. And we know that DNA is a complementary molecule. It has one side that complements the other, right? A to T, C to G. This structural property of DNA is the basis for a scary looking word, but nucleic acid hybridization. All that essentially means is that you have base pairing of one strand of the nucleic acid to a complementary sequence on the other strand of the nucleic acid, and that's really all it is. This nucleic acid hybridization forms the foundation of virtually every technique that is used in genetic engineering. Genetic engineering has become important in a lot of different fields. We see it right with food. We've seen it in forensics. We see it when we're doing gene therapy, right? It's everywhere. And the basis of the DNA molecule is important when we're talking about the genetic engineering stuff. So when biologists or geneticists, whoever, forensic scientists, whoever it is working with DNA, they face a couple of challenges, particularly because DNA, although it doesn't seem big to us, it's actually a very long molecule. And a single molecule carries, we know, thousands of genes, right? Usually, or generally speaking, a person who is studying a piece of DNA only wants to look at a certain component or a certain gene, certain sequence, a certain segment of that DNA. And we know from our introns and exon stuff that there are some parts of the DNA that are non-coding sections. So biologists, geneticists, whoever, molecular biologists, they really want to get in and study that one particular area that they're looking at, one particular gene, and get rid of all the other stuff that they don't want to look at. So to work with specific genes, scientists have developed methods for preparing well-defined segments of the DNA and then creating copies of them in this process called DNA cloning. So most methods for DNA cloning are going to be using plasmids. And you remember plasmids, right? You heard that in the lecture I did earlier today. Plasmids are pieces of DNA that are small pieces of DNA that are circular in shape. Remember those pictures I showed you? That replicate separately from a bacterial chromosome. We know we find plasmids inside of chromosomes. So to clone pieces of DNA in the laboratory, researchers will take a piece of a plasmid. This is usually taken from a bacterial cell, and they will genetically engineer it for efficient cloning. What they do is they take the piece of DNA that they're trying to study and they combine it with the plasmid. And when they do this, now you have what you call recombinant DNA. Recombinant DNA is the plasmid taken from the bacterial cell and the DNA that they want to study combined with one another. They then reinsert that DNA into the bacterial cell again, where it will be replicated over and over again. And then they're able to withdraw those genes, and now they have multiple copies of that gene that they needed replicated in the first place. That's the process of gene cloning. And the second kind of part to this is not only do you need multiple copies of the gene that you're trying to look at, but you need to specifically get to that piece of DNA, that specific gene that you want to look at, whatever it is. So biologists use restriction enzymes, and there's an animation here, but I will come back to that shortly. We just talked about gene cloning. So restriction enzymes. Restriction enzymes were discovered in bacterial cells in the 1960s. These are enzymes that are only found in bacteria, but they are actually designed to cut up DNA. They're in bacteria because they're like a protection for bacteria. They help to chop up any foreign DNA, particularly from bacteriophages that enters them. But we were able to take them, remove them, and use them to cut up DNA for analysis and genetic engineering. So they have specific areas of DNA that they look for and cut like a pair of scissors. So I'm going to go back to this animation, and I want to show you how restriction enzymes work.
Restriction enzymes are enzymes isolated from bacteria that recognize specific sequences in DNA and then cut the DNA to produce fragments called restriction fragments. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAATTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT. This restriction enzyme recognizes the DNA sequence CTGCAG. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence ACGT. This restriction enzyme recognizes the DNA sequence CCCGGG. It cuts the DNA strands between the bases C and G. The resulting DNA fragments have no single-stranded sticky ends.
So if we were trying to isolate a certain gene that we wanted to look at for some sort of genetic engineering, we would use restriction enzymes that are designed to specifically cut certain areas of the DNA and cut out that piece of the DNA or that gene that we wanted to analyze so that we could get rid of the rest of the stuff, if you will. So that's what restriction enzymes are used for. This process of gene cloning that I was talking about, sorry, I was looking at what was on the screen. This is an overview of the process that I described, how they use the plasmid and the gene of the chromosome that they want to, or the gene on the DNA that they want to look at, and then some of the uses that we have for it. Like I mentioned before, we use this for looking at gene therapy, looking at, crops and agriculture. In this case, a gene used to alter bacteria for cleaning up toxic waste, any, human growth therapy or gene therapy. Forensics is another one. If you get a copy of a gene or DNA, a piece of DNA from a crime scene, and you want to make sure you have enough of that sample so that you don't lose it, things of that nature. All right, so that's gene cloning, and we started to talk about restriction enzymes. So in the video, they showed you how restriction enzymes work. They're all different. There's all different kinds of restriction enzymes that cut in all different places. So normally you're using different types. In this particular picture, no, they're not showing you. But in this particular picture, they're using a restriction enzyme called EcoR1. It's a pretty popular one. It's designed to specifically cut any section of the DNA that looks like this sequence, GAATTC, like they described in the video. So that's going to create this cut up version of the DNA so that you can analyze specific portions of it. So what's the next step once you have your pieces of the DNA, or your restriction fragments, they call them? You're going to use that in a process usually called gel electrophoresis. Now, if we were in school, we would do this together, but gel electrophoresis is all about separating DNA out by its different size fragments. So this is what a gel electrophoresis chamber looks like. It's essentially a box that has this jello-like stuff in it. It kind of looks like agar, if you guys remember the agar cubes earlier in the year with the different sizes. And you use a comb. It has little spokes on it, and you set it, and then it sets like jello, and it becomes sort of a gelatinous, somewhat solidified substance, and it has these holes in it, or these wells. You remove the comb out when you're all done. And then you insert your DNA samples into the wells, and you hook up the box to an electrical source. So because DNA is a negatively charged molecule, when you put it inside the wells and you hook it up to the negatively charged part of the power source, the DNA is repelled from that negative charge, and it will travel through the wells, through the gel, moving towards the positive side. And when you're done, you get a DNA fingerprint. And a fingerprint, not like the ones on the tips of your fingers, but something that looks like this. These are all the fragments of DNA that were separated out by your restriction enzymes, and they are separated by size. So if you're imagining they started here at the wells and they're moving through to the other side, the ones that are bigger and larger are going to take more time to run through the gel, so they're not going to make it as far as ones that are smaller. And you can use this in comparing DNA samples. This is used a lot in forensic science. If you have an unknown sample of DNA and then you have, say, like suspects, family members, things of that nature, you're able to create a DNA fingerprint or the restriction fragments, and then you'd be able to compare, and if they have the same banding patterns, you would know that they were matches. So this is one thing that you would use, or use your restriction fragments that you cut up with the restriction enzymes to give you these bands, and then you can use that to determine relatedness, things of that nature. All right, and then finally here on our tour of biotechnology, I would encourage you to read the very couple pages that there are in the textbook on this stuff. Is this process of amplifying the DNA, or what we call PCR. So PCR is able to produce many versions of a DNA piece that you're using. This is very popular in forensic science because if you get a blood sample from a lab, you want to run it through PCR and make many copies of it so that you have you don't lose it, right? If something happens to that sample, you're SOL, right, as they say. It stands for bad word, out of luck. You can't go back and get more blood, right? So this is something that is done. A technique that is used to make billions of copies of a specific target of a DNA sequence. The PCR procedure is a 3-step cycle that brings about a chain reaction that produces an exponentially growing population of identical DNA molecules. So it looks like this. The first thing that happens is you take the sequence of the DNA that you're studying and you denature it, which essentially means exposing it to heat, and that causes the 2 strands of the DNA to denature or come apart from one another. Annealing to cool them, and you put primers on them just like you would in DNA replication. And that's going to form the beginning of what will be the new pieces of DNA that are made. So you're able to yield more copies of that molecule over and over again until you have many copies of the DNA. So I'm going to show you a video of PCR because I don't think this picture does a great job explaining how it works. Here's the steps for you if you go back and look at the notes. This is just talking about how you get more copies. So let me show you the video. I think it does a better job.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
All right, so that is PCR and how it works. You don't need to know all the crazy steps there. Really all you need to focus on is, or just know essentially that it's just a process that makes more copies of DNA. So that's what that's all about. On Thursday, you're going to be doing a virtual version of a gel electrophoresis lab. You're going to go through and look at how the restriction enzymes work. You're going to be loading a virtual gel, looking at the fragments and how you would analyze them, how you would measure them, things of that nature. So you're going to go through and look at the actual techniques that were described in this lecture. All right, so thank you so much. I will see you soon, maybe. Take care, everybody.